Hey, how's it going? This is the MERG. Today we're discussing active transport through the cell membrane. Active transport results in a molecule moving from an area of lower concentration to an area of higher concentration. One example is the heavy concentration of iodine ions in the cells of the thyroid gland, or the sugar that is almost completely absorbed from the gut by the cells that line the intestines in the digestive tract, or the water that is gathered up by the kidneys using active transport of sodium ions by cells lining the kidneys. Active transport requires a protein carrier and cellular energy in the form of ATP. Sometimes you'll hear people call these protein carriers pumps, and it works very similar to pumps. The energy obtained from ATP is used to move substances against their concentration gradient. A very popular active pump moves sodium ions out while pulling potassium ions into the cell. This process begins as sodium found inside the cell bind to the protein transport. ATP releases a phosphate group, becoming something we call ADP, supplying the transport protein with its connected sodium ions to mutate in a way that opens up the gate to the outside. Sodium ions are then released to the environment. Afterwards, potassium now jumps into the transport pump to replace the depleted sodium. This activates the signal to release the phosphate group that supplies the power to mutate the transport protein back to normal, releasing the potassium inwards into the cell. It's kind of an exchange that happens. I'll give you 3 sodium ions while you give me 2 potassium ions. The cells like to keep a high concentration of potassium ions inside by using sodium ions. This is very useful for your nerves and your muscle cells. Another important pump deals with the passage of sodium chloride, also known as salt. Sodium ions are pumped through the membrane into the cell, then chloride ions diffuse through the appropriate channels after. In cystic fibrosis, a mutation of these chloride ion channels causes them to malfunction. There is also such a thing known as bulk transport. As the name implies, this is how larger molecules like polysaccharides and polypeptides get moved across the cell membrane. Now listen to this. Bulk transport uses vesicles rather than transport proteins and channels. It's a process we call endocytosis. A small portion of the membrane invaginates or cups inward and forms this pouch with large molecules inside of it. That pouch pinches off to form what we call an endocytic vesicle, which will travel to its destination inside the cell. White blood cells are famous for this. They take up disease-causing pathogens in this exact same method we call phagocytosis. Phago meaning to eat. Another term, penocytosis, peno meaning to drink, is when the same thing happens but the cell takes in fluid molecules. One other term you should know is exocytosis. It's the opposite, where the already formed vesicle connects and fuses to the membrane from the inside and opens itself to the environment to secrete substances such as waste or other products. Take neurotransmitters, for example. These are larger signaling molecules that leave one nerve cell to excite the next nerve cell or muscle cell. There are also receptors found on the surface of the cell membrane, picking and choosing specific molecules of interest for endocytosis. This process is called receptor mediated endocytosis. Some people with a genetic disorder that fails to signal this type of endocytosis can sometimes lead to symptoms of cardiovascular disease. Their cells will fail to identify and therefore fail to take in important lipoprotein and cholesterol molecules from the blood. Well, there you have it. If you like my video, please like, subscribe, and share with your friends. If you have any questions, please comment below. I'll try my best to answer anything I can. Bye.